is how we're making these gametes, right? I'm going to pull my parents. If the question told us we're heterozygous, right, for both genes. And remember, we want to use that FOIL method. So I'm going to take my first B, my big B, combine it with my big E. I'm going to take my big B, combine it with my little e. I'm going to take my big B, my second B, very tired, combine it with my big E, you see here, right, and take my little b and combine it with my little e, which is what we see here, right, and that's one parent. Okay, so we're taking mom's genotype. We're asking ourselves, what are all of the different haploid gametes? Right, so these are all haploid. I have one of each allele, one of each gene in there. I have a B and an E. Okay, then I would do the same thing with dad. Okay, now in this case, dad happens to be the same. He has the same genotype, so he's going to make the same gametes, but that wouldn't necessarily be the case. Right? Otherwise, everything's going to be the same. Remember when we set up these boxes, the rule was one gamete above each box. And since we've made so much work to make gametes here, it just involves putting one egg above each of our Punnett squares across the top. Remember, ladies go across the top. And one sperm next to each Punnett square going down the side. Okay, so fundamentally, this setup hasn't changed any, it's just bigger. Pardon me, than the Punnett squares that we've set up for previous problems. So instead of having a 2 by 2, right, we have a 4 by 4, or 16 total squares. And remember then, next step, what we want to do is do fertilization. So you're going to drop the eggs and the sperm into each block. Okay, and you're going to keep like letters together, right, because that's going to make those letters translatable. So I'm going to do my 2 B's, then my 2 E's. And all of our other rules still apply, right? You always want to write the dominant letter first. You see we've done here, so that's going to make it easier to translate later. Okay, so again, even though this is bigger, we've done everything the same that we've done with our teeny squares. So then our last step is going to be actually translating the phenotypes that are in here. Right, so remember my question was, what % golden am I going to get? So remember, this is an epistatic problem, where the second gene turns the first gene on and off, right, like unplugging that oven. Right, so any box that has a capital E in it, right, and that's all we need is one dominant E. Okay, we're going to plug in that oven, means we're going to make color. I'm sharing my screen, right? That's not what all those pigs are about?
Yeah.
Okay. Okay, so anything with a capital or dominant E, I'm going to make color, which means I'm actually going to bother to look at what those B's are. All right, so you see then, with those, we can look at the B's, and remember, if we have a dominant B, we end up with black labs. Okay, and if we are homozygous recessive, we end up with chocolate labs. And I've been lazy, and these are just black and brown dogs. For the subset that do not have a dominant E, okay, these here, and there are 4 of them. We need to be careful because these are not always next to each other, right? But when we don't have a dominant E, remember this is like unplugging the oven, right? We've turned off our color gene. Once we've turned off that color gene, I don't give a hoot and a holler what it is, right? Because I'm turning it off. So we can literally ignore or cross out whatever comes before it because it's never going to get to do anything. Okay, these are what make our white dogs, or golden, because that's what makes them sell in pet stores. Okay, once we find these boxes, right, with the recessive E's, then it just becomes a game of counting how many we have. So I have 1, 2, 3, 4 boxes out of 16 total, which gives me that 25%. I'm operating under the assumption that you held up your end of the bargain, at least tried this while we were apart. So any questions about how we all got from here we have 2 puppies to here we have many puppy bebés? Okay. Most people, when I glanced at the Socratic, although there were a few people trying it last night, I think there's like 5 or 6 of you, most people were nailing this first question. The second question is, of course, much harder. It's designed that way. I kind of wrapped up as many different things as I could think of in one question. So if you don't get it, don't be disheartened. First off, no test question is going to be that hard. But if you can get all the different moving pieces in a question like that, you should feel good, right? Very confident about your ability to work through these types of problems. Because that one has, like, I think 3 different types of things you have to think about. And I saw a few of you were getting that one too. So you guys are doing really great with this. Plus, I think genetic problems are kind of fun. So you're going to study some way, right? Remember, we want to do active studying. So whether it's redoing your in-class activity problems fresh, right? Redoing some of the practice problems from lab fresh, right? Write them down and try them. Whatever it is, rewriting our notes, okay? Anything active is a much better way of studying than just reading, right? Our brains get bored very easily, and the second they get bored, they're not really logging any information anymore. Okay, any questions about this or any of the problems we've set up? Anything so far? Just cut it that way. We're going to get anywhere otherwise. All right, bye-bye puppies. Bye-bye puppies. I said bye-bye. All right, as I mentioned, I'm cutting a lot from genetics. So the only other thing, in fact, that I want to talk about is this definition, which is if you're using the class notes, you're just going to skip that genealogy stuff, the stuff that looks like family trees or pedigrees. Okay, so we already talked about Mendel's principle of segregation, right, which was up here. And remember, that was talking about taking our alleles, right, or basically our sister chromatids that has alleles on them, and how each one is segregated, right, into its own individual germ cell or gamete, right? And that when we did that, right, we didn't tear these alleles up into tiny pieces of confetti. Okay. The principle of independent assortment then says that when we are doing this, Okay, this movement of sister chromatids, right, from the parent cell to the daughter cells, right, to those gametes. Okay, that separation happens independently. Okay, or as we think about the two ways we get this independence, right, in a way that allows for a lot of random mixing, okay, or a lot of diversity. Okay, these were all the things we talked about that cause diversity when we were doing meiosis, right? We really focused on these in like meiosis 1 in particular. How do we increase diversity? Okay, so for example, right, when we do metaphase, remember there isn't a mom side and a dad side of metaphase, right? So when they all line up in the middle, it's random which way they're lined up. Okay, so when they're pulled apart in anaphase, each side of the cell is going to have a random subset of some of mom's and some of dad's copies. Okay, in addition, don't forget, right, we had crossing over happening. Okay, so what all of these things say, okay, is that as our alleles are moving around and being shuffled into their final gametes, right, different genes, okay, the genes for hair color and the genes for height, are not necessarily moving around together, right? Okay, they move around completely independent of one another. Okay, so whether it's because they get separated potentially by being on different sides of that middle line, or maybe they get separated because one stays on the chromosome and the other one gets switched during crossing over, right? There's lots of different ways in which we can continuously get mixed up. So while the principle of segregation says a single gene, right, a single allele isn't broken up and torn up into bits of confetti, right? We don't want to tear up how to make hair color. Still need to be able to do that, right? Two different genes, right, hair color and height, are not necessarily going to stay together, right? We can pull those apart and mix them up, thanks to things like crossing over. If I switch mittens with Jasmine, right, that mitten may have how to make height, but not, right, how to do hair color. That might stay on me. That's my boots or something. So independent assortment is our diversity. Now, any questions about this? This is going to finish our genetics. We're not going to do anything else that's in there. All right, so basically our main focus on genetics were some of our vocab and definitions, then of course our Punnett squares. I didn't cut any of that stuff. Alrighty. Jasmine, can you stop and then once we get going and restart?